Okay, we're back for the second uh, lecture on cells. So this will be 203 cells 2. This is where we left off, a representation of the cell membrane, the phospholipid bilayers, cholesterol. Among the phospholipids, those blue things are membrane proteins. There could be channels, cell ID proteins, sometimes enzymes, sometimes receptor proteins. And then there are filaments of cytoskeleton underneath. These are protein fibers that give cells their shape. So an integral protein is one that goes across the entire width of the cell membrane. Then a peripheral protein, so it's going to be a protein on one side of the membrane. And those green things are cell membrane proteins, are glycoproteins. That means they have some sugars attached to them. And there's the functions. Physical barrier establishes a flexible boundary, protects cell contents, supports cell structures, and separates two aqueous solutions from each other. Selective permeability, 2, regulates entry and exit of ion, nutrients, waste products through the membrane. Regulates what can cross the membrane and when. Electrochemical gradients. This is in concert with selective permeability because establishes and maintains electrical charge difference across the plasma membrane. Because there's a slight difference in charge across the membrane, slightly more negative inside than outside. And this is due to a disequilibrium of ions across the membrane. It's absolutely critical for nerve function and muscle function, although it's true for every cell in the body, is slightly more negative inside and outside. And that's maintained by the selective permeability of the membrane and active transport. The communication contains receptors that recognize and respond to molecular signals. So ligand receptor signaling. A ligand is just something that binds the receptor, all right? So it's important for cell-to-cell communication. So there are signaling molecules that are sent out. They're called various things. In the endocrine system, we're going to talk to, call them hormones. In the nervous system, we're going to call them neurotransmitters. It all depends on how far they travel to bind to their receptor. So it's cell-to-cell communication. Channel length or chemically gated channels. So here's a thing. A gated channel has a door on it that is closed. This ion or molecule can't cross if it's closed. It's gated. And there are certain things that open gates. And we'll talk a lot more about this in the nervous system. We'll talk about chemically gated. Things bind to a receptor associated with the channel that opens the channel and allows this substance to cross. Right? There are other things that can open channels also. We'll look mostly at mechanically gated channels also, and what we call voltage gated channels. And so these gates are related to channels, and mostly we look at them in what's called postsynaptic membranes, and membranes that are on the other side of the synapse. We're going to get to that in the nervous system. Enzymatic receptors. There are actually enzymes in the cell membrane, particularly in the digestive system, that can get turned on by proteins, and there are peptide hormones that can turn on enzymes inside the cell when they bind to a protein receptor on the outside. G protein coupled reactions, second messenger systems. as I mentioned before, if the signaling molecule is water soluble, it doesn't get into the cell. It binds to a receptor on the surface of the receiving cell, and this activates what's called a G protein just underneath the surface of the cell. This will be a peripheral protein. And it turns on enzymes, similar to the enzymatic receptors turns on enzymes that makes a second messenger. And the second messenger goes throughout the cell and turns on all kinds of stuff inside the cell. Oh, and kinases are enzymes that add phosphates, phosphorus groups, to proteins. And that generally turns these proteins on when you add a phosphate to them. That's called a kinase. And adrenergic synapses. These are synapses that use norepinephrine. We're getting a little ahead of ourselves now. In the nervous system, we'll talk more about these. How adrenergic uses norepinephrine, and they use second messenger systems. Whereas cholinergic synapses that bind acetylcholine do not use second messenger systems. So we'll get to that later on. So here we go, channel linked receptors. So you have a ligand, see this channel is closed here, a ligand binds to it right here, so there's a receptor for the ligand on the channel. It opens up the channel, allows ions to cross the membrane. Here's enzymatic receptors, inactive protein kinase below the receptor. The ligand binds to the receptor. It turns on a kinase that adds a phosphate to a protein, and this protein is turned on. Then G protein coupled receptors, ligand binds the receptor, turns on a G protein makes a second messenger, which is often a molecule called cystic AMP. And the second messenger goes on, activates kinases and other things, turn on proteins all over the cell, and causes the cell to make changes. Sometimes it opens up ion channels. That would be your adrenergic receptors. Sometimes it actually goes into the nucleus and turns on genes and causes certain proteins to be translated. Okay, organelles. So let's talk about the organelles in the cells. And what you need to know about this is just the name of the organelle and generally what it does. Generally, what is its role? So the nucleus has a nuclear envelope. It's actually a double membrane. It has big holes in them. They're called nuclear pores that allows big molecules to leave. In the next part of the chapter, we'll look at messenger RNA molecules that have to come out of the nucleus, and they're big. So you need a big opening for them to go through. Mitochondria, the powerhouse of the cell. Not all ATP is made inside mitochondria, but the vast majority of ATP is made in mitochondria. So that's what we call the powerhouse. By the way, I have endosymbiosis next to mitochondria. Ignore that. Okay, lysosome, proteasome. These are similar things. Lysosome are recycling centers. Organelles that break, are taken or malfunction are brought in lysosomes, and there's proteolytic enzymes in there that chew up the organelle that's used up and throws away waste products and saves the important stuff. Proteasomes is almost the same thing. They chew up protein complexes. There's proteases inside which chew up proteins. Very similar things. Another similar thing is called a peroxide. So we have digestive enzymes inside there, and these are detoxifying. So any kind of foreign molecules, including drugs that you might take, or just in your food, these are sent into peroxisomes. I mean, there's certain toxic molecules that actually we eat all the time, and these are chewed up and destroyed by peroxisomes, mostly in liver cells. Liver cells have a lot of peroxisomes. So if you eat something that's really toxic or dangerous, the liver will take it in, put it into peroxisomes, chew it up, and make it innocuous. So those are all membrane bounded. Ribosomes are not membrane bounded, and that's where mRNA is translated, and you make proteins according to the message in a gene. This is cytoskeleton. Mostly we'll look at things called microtubules that allow things to be moved inside cells, particularly chromosomes and cell division. And the cytoskeleton gives, there's several types of cytoskeleton fibers. We don't have to know all the types. And they give the shape to cells, and also assist sometimes in exocytosis and endocytosis. Centrosome with centrioles. These are microtubule manufacturing areas in the two poles of the cell. And they are mostly used during cell division when you have to, when you replicate your chromosomes, and during mitosis, you attach these microtubules to chromosomes and you yank and pull chromosomes to different parts of the cell. That's the centrioles are in a centrosome, and that's where microtubules are constructed. On the cell surface, cilia and flagella, cell hairs, they also have microtubules inside of them. And cilia are small cell hairs, flagella are longer cell hairs. And we can tell what kind. Okay, wait a second. So let's talk about cilia and flagella. One important place to find cilia is lining your respiratory passageways. Respiratory passageways is a mucous membrane. There are certain cells called goblet cells that make mucus. And so when you breathe in air, but you breathe in a lot of other stuff too. Dust, bacterial spores, fungal spores, all kinds of stuff that's been floating in the air. And you don't want these things to reach these small areas, the little blinded sacs in your lungs where gas exchange occurs. So what happens is this fungal spore gets down there and likely will hit the side of the lung passageway and gets stuck in the mucus. So now the cilia that are beating, and they bring that mucus up to your larynx, or in your nasal passages, the cilia bring mucus down to your larynx, and this mucus is put out into the what's called the laryngopharynx, where you can swallow it, and you can swallow the mucus. So everything this time you're swallowing mucus that's been brought up from your lungs that has all this stuff that's stuck in it. Of course, your mucus will go into your stomach, you'll digest your own mucus, and the stuff, all the stuff that you breathe in will be voided in feces. So it's a way of house cleaning, like vacuum cleaning the respiratory system using cilia. That's true for every human being. Female human beings also have a ciliate epithelium in fallopian tubes. When an oocyte is released, it's not mobile, so it has to be moved down the fallopian tubes by being cilia. So that's a ciliate epithelium, but only found in females. Now, flagella allows cell movement, and there's only one flagellated cell in all of human biology, and that's only made by one of the genders, males. Sperm are the only flagellated cell. It has a flagella and allows sperm to be mobile. And these are actually membrane-bounded organelles. Now, there's a flagella, if you go look into bacteria, you go take bacteriology, there's something called a flagella that some bacteria have. It's a very different thing. So it's unfortunate it's the same name. So eukaryotic flagella are quite different. They are actually membrane-bounded organelles. Okay, endoplasmic reticulum. This is folded internal membranes that are connected to the nucleus, actually. And there's two types. The rough endoplasmic reticulum that has ribosomes along it. And that's where dangerous proteins and proteins that are going to be exported from the cell are produced in this protected area inside this membrane. And then there's also the smooth endoplasmic reticulum where lipids are made. So rough manufactured dangerous and secretory proteins. Dangerous proteins would be proteases, enzymes that chew up proteins. These are made in an inactive state and have to be turned on when they're exotype, when they're pushed out of the cell. As smooth as lipid synthesis. Then a Golgi apparatus. Once a protein is made in the endoplasmic reticulum, it's sent to the Golgi apparatus, which is a stacked series of membranes, where the finishing touches are put in. The proteins are detailed. They're changed, final processing of proteins. And then they can be put out of the Golgi apparatus and either go into a lysosome, go into mitochondria, go into a secretory vesicle, go to the cell membrane, and secrete it into tissue fluid, like protein hormones. They're made in the ER, they're sent to the Golgi, and then pushed out by exocytosis into interstitial fluid, then to the bloodstream to go to distant places. So this is what's called part of the endomembrane system. Endo means internal, so it's an internal membrane system. And remember, that's the hallmark of the eukaryotic cell. Internal membrane, membrane-bounded organelles that prokaryotes do not have. And so the ER, the Golgi, the lysosomes, the peroxisomes, vesicles, transport vesicles, all part of the endomembrane system. Then cellular junctions. And these I didn't put up this slide, but these membrane junctions let's see. Yeah, right there on page 133, the membrane figure 432 with the membrane junctions. Tight junctions prevent leakage between cells. So you have some particularly like the GI tract where you don't want to have, you want things to go into your blood through cells, not between the cells. So they're called tight junctions to prevent leakage between cells. Then there are desmosomes, provide structural support. These are like spot welds. I like to think of them as spot welding, where they hold two cells together that are being moved. Like in your heart muscle. Actually, the intercalated discs between heart muscles is a good example of desmosomes, because remember, these things are contracting and they're moving, and you don't want them to tear apart. So they have to be held tightly together, and desmosomes do that. Then gap junctions. These are electrical connections between cells, little holes that allow the cytosol of one cell to be directly electrically in contact with the cytosol of the cell right next to it. And what the electrical connection means is ions can travel between cells directly, because those are the vehicles of bioelectricity, ions. So ions can travel directly between cells through these gap junctions. And these are between heart muscle cells also. So one heart muscle cell can directly stimulate its neighboring cell to fire an X potential and get excited. Get excited and then fire an X potential. So they also found a little bit in the nervous system. They're really also called electrical synapses. When we get into the nervous system, we'll learn that most synapses are called chemical synapses, where there isn't a direct connection between the two cells. They're very close, but they're not directly linked. But in some cases, there are direct connections between nerve cells, and ions can cross the membrane directly. So that's either gap junction or electrical synapse. And that's it, folks, for this part of chapter 4. So I'm going to call it right now and end this, and I'll see you on the next when we start talking about the other part of chapter 4 on gene function. So I'm going to start the recording now.